Good afternoon. Thank you so much for coming for this first talk on the gut microbiome, its effects on health. This is a topic that is a topic of research of this century because the whole field has developed just in the last few years. I first heard about an effect of the microbiome. Microbiome are the bacteria, the viruses, the fungi that live in the gut of all of us. And the numbers of these are way, way higher than the number of cells in the human body, several folds higher. So they do have an effect on health. They may be just living there, symbiotic, having no just beneficial effects on both sides to each other, having no effect at all, or harming the host. And although generally they will live peacefully with the human in the human gut, they can get disrupted, and that is by disease, diet, medications such as antibiotics that are so commonly used. But anyway, this field, the first time I heard about anything about microbiome or the bacteria was when there was a report on transplanting fecal matter from one mouse to another mouse. And what was done was to take fecal matter from a lean mouse, a thin mouse, and put it into the gut of an obese mouse. And the obese mouse started to lose weight. So obviously there was something in the bacteria that were doing it. This fecal microbiota transplant has now gone on to actually become a treatment choice. And I was reading that it is now used for fecal matter transplant, which can be done by colonoscopy, by enema, by capsules, by suppositories, and it is a treatment of choice for diseases such as inflammatory bowel syndrome, colitis, constipation, and neurological diseases also, such as multiple sclerosis and Parkinson's. So the gut-brain axis is now called the gut microbiome brain axis. And it is known to influence also the adrenal function and stress response. This was also a very early study which showed that mice that were brought up in a germ-free environment responded to stress much more than those in the germ who had the microbiome. So obviously there was something there. These early studies were done in about 2002, 2004, and since then the field has really exploded. And we now know that the microbiome is involved in a host of human function. And I'm hoping that this series of 6 lectures will cover some of these, not all of them. Today's talk will cover the effects of this on Parkinson's disease. And then later on, we will have other topics covered that I will talk about later. But it is known to affect normal function as well as disease states, diabetes, cancer, and definitely brain function, nervous function. Today's speaker is Dr. Malu Gomez-Tansey. She is a professor, the Norman and what, Susan? Fixel Professor Chair in Neuroscience and Neurology at the University of Florida. And the Fixel, as you know, is located next to us. Dr. Tansey. Dr. Tansey has worked at Stanford and got her training at Stanford and the University of Texas at Southwestern Medical Center in Dallas. She has worked at Emory University, and in 2019, she was recruited here to serve as the director of the Center for Translational Research in Neurodegenerative Diseases. She's the first endowed Norman and Susan Fixel Chair in Neuroscience and Neurology at the University of Florida. She's working on oh, I didn't say, she's invented or discovered TNF inhibitors, TNF, that is tumor necrosis factor inhibitors, which have now been used in clinical trials in Alzheimer's disease and perhaps in Parkinson's disease. So a most accomplished researcher. She is a Ph.D. and is wonderful to deal with, in inviting her to come here. And I'm really pleased to have Dr. Tansey come and talk on Parkinson's and the microbiome. Thank you.
Thank you, Pushpa. Okay. It's wonderful to be here. Thank you for the invitation to kick off this really exciting course. I am going to try to put the information that Pushpa gave you into context of how we're thinking about the gut microbiome in terms of how it affects your lifetime risk for neurological disease, and in particular for Parkinson's. So I have a couple of disclosures with collaborations, consultants, physicians, advisory boards that I am required to tell you about. And one of the things that Pushpa mentioned is the TNF inhibitor discovery. Before I set up my lab in academia, I spent some time in the biotech sector in California and was fortunate enough to be given a project that had been on the shelf for quite some time. And it turned out to be a really great story that we were able to publish in Science in 2003. And today, 20 years later, those inhibitors are in clinical trials for Alzheimer's, mild cognitive impairment, and they were in trials for COVID-19. So hopefully with a little bit more work, we can make some justification for why they can be potentially useful for anything with chronological, with chronic inflammation. So the questions that I want to focus on today are shown here. I'm going to talk a little bit about what causes brain inflammation. Is there evidence of inflammation in Parkinson's? Are there certain types of cells or proteins, genes, or hormones that might contribute to this inflammation? How does good gut health benefit us? And what is the best way to keep inflammation in check? So this schematic is only to make the point that everything in your body is really interconnected, okay? The brain is not immune privileged. The brain senses everything that's going on in the external organs, and the organs are much of the time being told what to do by the brain, and now we know by the gut bacteria. So we know that things like interleukins, TNF, and other inflammatory factors are very important for communication between cells. So we can't completely get rid of them. What we hope to do is identify instances in which these chemicals are in excess that is causing inflammation, and be able to reduce their levels so that you don't have the pathology associated with chronic inflammation. So one of the things we know that because of infectious disease, such as COVID-19, there has been strong evolutionary pressure for us to develop genes and gene variants that give you a very strong and robust immune response. That's to keep you healthy and get you to reproductive age so you can pass on your genes. Now, we've lived too long, and one of the things that happens now is that the good genes or robust immune response genes may now be working against us. Why? Because one of the things that happens over a lifetime is that you're exposed to a lot of things in your environment. You have repeated infections, and all those things tend to come together and increase the inflammation in your body. And sometimes those immune cells, just like your neurons, are a little bit tired and they're getting old, right? So one of the things that we consider is, does this phenomenon of inflammaging, which is inflammation as you are getting older, this gives you low-grade chronic inflammation, right? It's not a fever, it's just a little bit too much inflammatory factor circulating around in your plasma. And something called immunosenescence, which is basically aging of the immune system, and the immune cells aren't able to do their job like they once did. The reason that aging of the immune system is so important is because age is the #1 risk factor for 2 of the diseases we study, and those are Parkinson's and Alzheimer's. So we know that individuals with Parkinson's can lead very, very active lives, shown here on the far left, and they also experience things like on time and off time when they're on medications. We know that some of them have tremor, but not everybody does. Many of them have this rigidity, right? The shuffling gait. But we now know that Parkinson's is probably not one disease, it's a lot of them. And it's really people living with Parkinson's instead of Parkinson's disease. Now, one of the things we know is that in the brain, just like when Alzheimer's has to deal with amyloid plaques and senile plaques, Parkinson's has the equivalent, right? And it's not called amyloid beta, it's called synuclein. And synuclein is one of these sort of badly behaved proteins that likes to aggregate and glum up. And it's been thought that this aggregation of this protein as you get older in certain brain regions are basically able to interfere with the function of your electrical impulses in the brain. But there was a discovery that was made by people looking at brains that basically revealed that it doesn't appear in all the brain areas at the same time. It appears to potentially start outside the brain, maybe in the gut, because your enteric neurons in your gut nervous system has this same protein, right? All neurons have this synuclein protein. And so by looking at a whole bunch of brains, neuropathologists decided that perhaps the pattern of deposition of the synuclein suggested that the disease didn't start in the brain, in the dopaminergic neurons that are so important for controlling movement. And that was coupled with the observation that many people, as they get older, right, eventually you lose your dopamine producing neurons. And when you cross that threshold, you get your motor symptoms, your tremor, and you get your diagnosis in a neurologist's office. But decades before that, as early as 2 decades, it is now thought that the disease is when that starts, okay? And it takes a long, long time to develop. And so my lab has decided to focus not on what happens after you have your motor diagnosis, but what happens in the stages that we call prodromal, or early stages prior to your clinical diagnosis, right? And it turns out that some of these things are impaired olfaction, urogenital dysfunction, visual impairment, GI dysfunction, and sleep disorders. And so these come as early as 2 decades before your motor tremor symptoms. And they're thought to be related to the aggregation of this synuclein protein in organs that control those activities, okay? So what we think about Parkinson's then is that it's a perfect storm of aging, plus the genes that you were born with, strong or lazy immune genes and genes that can combat environmental exposures, as well as the environment that you live in, lifestyle choices, diet, pesticides, pollution. So these 3 combine to give you your lifetime risk for these age-related diseases like Parkinson's and Alzheimer's. And by focusing on the earliest stages, we hope to understand how the immune system is sort of the first responder, the weather vane of what's to come. And by looking in your blood and by looking in your gut, we hope to identify whether you're at higher risk and then hope to slow down the development so that you never get the motor symptoms. So we know that environmental factors can disrupt the immune system. We know that they can alter immune cell function directly. We know that they can also cause some kind of an autoimmune response in you by triggering the production of antibodies that are destroying your own tissues. And we know that environmental factors can also directly affect tissues like pesticides affecting the gut microbiome, the absorption, etc. And so one of the things we have come to understand is that these environmental factors are basically, many of them, causing inflammation outside your brain. And with many, many years, that translates into brain inflammation or neuroinflammation, okay? And an increase in neuroinflammation in your brain, it's not huge inflammation, it's low-grade inflammation. It's like your brain doesn't even know that it's happening. That tends to accelerate the rate of neuronal loss, not in all parts of the brain, but in certain parts of the brain where neurons are making dopamine for a living, okay? So they're under a lot of oxidative stress, right? This is why antioxidants have worked really well in the lab, right? We've cured Parkinson's disease in mice many times over, but that's because we can give them antioxidants, anti-inflammatories, way before they lose any neurons, right? But how do we do that? How do we identify, what the processes are that are causing the neuronal loss? So I mentioned protein aggregation. Many of the diseases that we work with, Alzheimer's, Parkinson's, Huntington's, and ALS, they're all called proteinopathies because they're all in some way, shape, or form associated with aggregation of badly behaved protein, okay? And about 5% of people with these diseases have a genetic mutation, and the other 95 do not. It's what we call sporadic or idiopathic, okay? That doesn't mean it's not heritable, because genes are heritable, and you are looking at data that suggests that Parkinson's and Alzheimer's and these diseases cluster in families. But there are people who have very rare mutations that cause overproduction of the proteins at the very top, amyloid, synuclein, Huntington, oxidized protein. And that serves as a trigger to the immune cells in the brain, called microglia, to start producing inflammatory factors, right? Tumor necrosis factor, interleukins, IL-6, and that bombards neurons with cell death pathways. Now, the $64 million question is, how can inflammation, as a result of these protein aggregates, give you such different diseases, right? And we don't know that. We don't understand it. We think that there's different vulnerability in different parts of your brain, and depending on your genetics and the environment, you will develop one disease and not another. But we do think that inflammation is kind of a common mechanism for all of them. Now, what about the 90%, 95% of people that don't have mutations in these proteins, okay? Do they get inflammation? And the answer is yes. We think that you can get to this inflammatory cycle through the things in the blue boxes, which is environmental exposures, chronic systemic inflammatory disease, maybe obesity, atherosclerosis, pesticides, physical inactivity, unhealthy diet. So all these things can make your body inflamed, and then your brain senses it. And after decades of this, you have an increased risk for development of these diseases. So this is a model that we are testing in my lab and many others, because what it does, the good news is that if these diseases don't start in the brain, they start outside the brain through inflammation in other organs, then maybe we can stop them, right? We don't have to get the drugs into the brain. We just have to figure out where these diseases start. So this is a summary slide that just tells you how we know that there's inflammation and immune dysfunction in Parkinson's. There's histological studies of brain autopsy, but those don't tell us how early it starts, right? There's PET imaging. Those tell us that the inflammation starts before you get a lot of neuronal loss. So now we're talking, right? Now there's a possibility that we can intervene with that inflammation and slow or prevent further development. The genetics tell us that there are genes that predispose you to give a very hyperinflammatory response whenever you get immune challenged, either with an infection, maybe COVID, maybe the flu. And we know that by looking at the blood, the plasma, the CSF, and now more recently the stool, we can identify who's inflamed. And finally, the epidemiology. So these are studies that look at groups of people that either have one disease or don't, or have a disease and are being treated for that disease, and their chances of developing X, okay? So I'm going to tell you a little bit about how the epidemiology is helping us understand how middle age inflammation may be contributing to these age-related diseases that we see when you're 60, 70, and 80. So here's the synuclein protein that I told you about, right? This synuclein protein misbehaves, it unfolds, and it forms these toxic aggregates. And eventually, we think that your body walls them off into what we call Lewy bodies. So the Lewy bodies at autopsy actually appear to indicate that it's your body's way of walling off toxic protein. And the neurons that can't do it are the neurons that died, okay? So we know that synuclein is one of the things that's found in the brain. But as I mentioned before, the autopsy studies seem to indicate that there's a progression of the synuclein aggregation, starting from very low in the brainstem, progressing all the way to the cortex, almost as if it was following some kind of anatomical connection. And based on the early non-motor symptoms, hyposmia, anxiety, depression, sleep, and GI dysfunction, it is thought that the synuclein starts to aggregate in your nose, in your olfactory bulb, right, in response to inflammation in the environment, or in the gut, okay? And so people started looking, is there any synuclein in the gut? And it turns out that there is. Again, we don't know if that was a consequence or a cause of the disease, but we do know that people with Parkinson's appear to have this aggregated protein in the gut as well as in the brain. So we proposed a model, and we basically said, okay, if the neurons in the gut have synuclein, and synuclein misbehaves in the gut 2 decades before Parkinson's, then what is causing the aggregation? Why is it doing this? And where does it go? And so our model is that with inflammation in the gut, and I'll tell you some examples, we propose that this synuclein gets passed on from the gut into the vagus nerve and all the way up to the brain. But it takes about 20 years. And we know that people that have had vagotomies for, say, ulcer intervention, before we knew that you could treat with antibiotics, they have a lower incidence of Parkinson's. Almost as if you cut the highway, right, cut the vagus, that may prevent the further development. We also think that the synuclein could get in the bloodstream, and it doesn't have to go through the vagus. It just goes through the blood across the blood-brain barrier, okay? So there's many ways that this could happen. But the question is there any evidence of inflammation in the gut? Is there any evidence that this highway from gastrointestinal to maybe centers that control depression and sleep to motor symptoms to then cognitive impairment, which is in the super late stages of Parkinson's, and not everybody experiences it. We think that this whole highway can be regulated by how much inflammation is in your body. And so my postdoc called it inflammation is the dark passenger of Parkinson's disease. And one of the things we want to look at then is, what is the gut doing? And on the left, you have a healthy gut. And this is the lumen of the gut, where there's microbiota, there's fungi, there's viruses, and there's bacteria, some of which are good for you because they help absorb the food. Now, below that are the epithelial cells, right? And they're the ones that are responsible for absorbing the nutrients. And down below them, you have a whole bunch of immune cells, right? And the immune cells are there in case any of the bacteria happen to leak in between the cells. That's when you have a leaky gut, right? And when that happens, the inflammatory immune cells go to town. They get very excited. And so we know from a lot of studies that inflammation in mice and in humans causes an increase in the levels of the synuclein protein. And when the levels are high, guess what it does? It aggregates and misbehaves. So we think what is happening is there's a signal from the gut microbiota down to the epithelial cells that makes them leaky. And then bacteria that shouldn't be down inside in the lamina propria are now there, and they're activating the immune cells. And this activates a sort of ongoing immune situation. Now, let's talk about the microbiome and what it is. Most of the microbiome that has received attention is the gut microbiome, right? But you have microbiomes anywhere that is in contact with the environment. So you have a mouth, pharynx, respiratory system microbiome. You have a skin microbiome. You have a urogenital tract microbiome. And you have, obviously, a gut stomach microbiome. Incredible numbers, trillions of bacteria, fungi, and viruses that keep you healthy and some that do not. We're going to focus on the gut microbiome because of the connection to gut and brain. And where does the gut microbiome come from? So a fetus is considered to be sterile when it's inside the mother. And the bacteria that's acquired at birth through the vaginal canal of the mother is what determines its initial microbiome, okay? Bacterial composition is highly variable person to person and throughout a person's life. And it's determined by your diet, your genetics, your immune system, and the environment that you're in. Stress can change your microbiome. No sleep can change your microbiome. Smoking changes your microbiome. So we know that when you take antibiotics, you wipe out your microbiome, and then you have to eat some things that will repopulate it, right? So most gut bacteria in the microbiota benefit you because they help you digest food. There's wound healing, and the lining of the intestine is shed on a regular basis, and it has to remodel, right? And antibiotics do save lives, but they destroy the microbiota. They leave space, if you will, for the more pathogenic bacteria that colonize and take over. And so chronic antibiotic use is no bueno. And we now know that. However, there are infections like Clostridium difficile. You may have heard of it. It's a life-threatening infection, and you can't get rid of it with antibiotics very easily. So what they do is they do a fecal microbiota transplant, like Pushpa mentioned. And if they don't do it, they can't save your life. So it's as simple as taking a fecal microbiota sample, put it in your intestine, let it sit there, and off you go. So now they've come a little bit more into the 21st century, and they don't have to do it with feces. They can do it with defined bacteria. But either way, if I had C. difficile, sign me up. So the gut microbiome has been demonstrated to play a role under these conditions. All of these, believe it or not. And Parkinson's disease is where I'm going to focus. But almost any ailment can be pinned on the gut microbiome. And bacteria have DNA in them that can be sequenced, that is different than ours. And this is how we know what bacteria you have in the gut or in the skin or anywhere. And we also know that they're making really important substances that don't act in the gut, but act in the brain, or they act in your liver, or they act in the blood. So there's this whole world of microbes that we did not appreciate for a long, long time. And so we have this triple axis, the brain, the blood, and your gut microbiota. And the gut microbiota is actually responsible for educating the immune cells in the brain, the microglia. So what is the evidence that Parkinson's individuals have a dysbiosis or microbiome that's out of whack? There's been like 18 or 20 studies now, and it does look like they have increased things like Akkermansia and lactobacillus, and they have decreased Prevotella, Roseburia, Blausia, and others. Now, why does this matter? Well, the species that appear to be increased appear to be opportunistic pathogens, okay? Now, people go, well, if you have Parkinson's, don't take lactobacillus. That would be silly. But we don't know why lactobacillus is up in Parkinson's. It could be protective. So don't let anybody tell you, don't take this, don't take that. You really need to talk to your doctor, and we really have no good evidence that any probiotic can slow down the disease. It may give you good gut health, and that's important. If you feel better, you're going to be more physically active, you're going to be happier. And so it's important to have good gut health, but rest assured that there is no good data whatsoever to support the use of expensive probiotics to treat any of these diseases. Now, those that are decreased are bacteria that make short-chain fatty acids, like butyrate and propionic acid. Why does that matter? Because those are anti-inflammatory. So they keep the inflammation in your gut low. And if you lose them, then you're probably going to be more inflamed. So we did a study at Emory where we looked at 156 Parkinson's patients, 110 controls, and 49 spouse pairs. And this is really important because if you compare someone with Parkinson's and someone with no Parkinson's in different households, they're going to be very different. So the spouse is a really good control because they're living in the same environment. And what we did here is we looked in the stool, okay, collecting it from a swab, from a toilet paper. We extracted the protein and we looked and asked, what are the different factors that are in Parkinson's and not in the spouse? And we found 4. IL-1 alpha, IL-1 beta, a chemokine called CXCL8, and C-reactive protein. And all these are driven by NF-kappa B, a very important pathway that causes inflammation. Now, we said, well, is this just in Northern Europeans, or is this just at Emory and UAB where we did the study? And so we confirmed that with a different cohort in Finland and a colleague of ours. And this time we collected blood as well, and we collected clinical metadata. And here's what we found. Parkinson's patients have lower butyrate, a short-chain fatty acid that's anti-inflammatory, and lower propionic acid compared to the controls. They also have higher calprotectin. What is calprotectin? Calprotectin is the quintessential biomarker for inflammatory bowel disease. What does this mean? This means that Parkinson's patients appear to have a leakier gut, okay? Gut inflammation. We don't know when it started. We don't know if it's a result or a cause of the disease. But here's some data that suggests that higher levels of the stool chain fatty acids and lower levels of the inflammation correlate with later onset of Parkinson's symptoms, suggesting that if you can keep your short chain fatty acids up, you may be able to delay your motor symptoms, okay? Now, this is just a correlation, and the clinical trials really need to be done to ask, does butyrate or propionic acid improve gut health, lower inflammation, and does it do anything to your motor symptoms? So, nutrition. What is the best diet intervention for good gut health if you have Parkinson's? I'm afraid that the answer is discuss with your physician. The reason is it's important to know what medications anybody's taking so that they don't interact poorly with whatever diet or probiotic you're taking. Carly Rush is a Ph.D. student in nutrition at UF. Hopefully she'll be able to come talk to you about diet. But she published a study where they showed that the Mediterranean diet was able to improve constipation symptoms in Parkinson's and might be able to reduce some of the non-motor symptoms that they had. And this was a 5-week pilot study here at UF. So the hypothesis then is that intestinal inflammation causes misbehaving alpha-synuclein and perhaps other proteins. They aggregate, they circulate, somehow they get into the brain. And what are we going to do about it? Okay. So what other evidence is there that gut inflammation may be related to increased risk for Parkinson's? Well, this is what happens in inflammatory bowel disease. Okay. You have certain genes that predispose you. You have certain environmental factors that also predispose you. And then what happens is that you get a loss of this epithelial barrier integrity and the bacteria leak in, okay? You get an expansion of the autoinflammatory processes. You get circulating antimicrobial antibodies, right? And that's a subclinical inflammatory state. And finally, when you get this uncontrolled immune response and bowel damage and tissue remodeling, you get your IBD diagnosis. And the reason that this is very similar to what we see in PD or anything is that if we could just stop the inflammation at the subclinical stage, you wouldn't ever get the IBD diagnosis, right? So that's the idea. We want to understand what are the earliest things that happen. So this is when I tell you the epidemiology also supports the idea that gut inflammation, chronic gut inflammation, may not be in your best interest. And these are studies that are small, so they need to be confirmed. They were done by a colleague of mine at Mount Sinai, Inga Peter. And basically, she found that patients with IBD are more likely to develop Parkinson's unless they are treated for their IBD with anti-tumor necrosis factor drugs. These are Enbrel, Remicade. You've seen the commercials on TV. They're used for a lot of autoimmune diseases like psoriasis, rheumatoid arthritis, all sorts of things. When do these diseases start? They don't start in your 50s and 60s. They start in your 30s and 40s. So what this data says is if you have IBD, you may have an increased risk. But if you're treated with an anti-inflammatory, your risk goes through the floor. So that's pretty exciting, I think, because it tells us that there's a window of opportunity during which we might be able to identify people at risk and we might be able to intervene with safe drugs so that they don't go on to develop the disabling motor symptoms. So what is TNF? It's called tumor necrosis factor. It is the granddaddy of inflammatory cytokines, okay? It's made by microglia, which are your immune cells. It's made by monocytes. It's made by all immune cells. And what people know is that it produces inflammation, right? If you have any kind of autoimmune disease that has chronic inflammation, you have an increase in soluble circulating TNF. But what people don't know is that it's also important to have in your body because it helps you fight infection. Why is that? Well, it's made as a protein that's attached to the cell membrane, okay? And in this membrane attached form, this protein helps you fight infection. If you don't have it, you're going to have all sorts of opportunistic infections. It's also important during development so that all your lymphoid organs, your bone marrow, everything develops well. It's not always attached to the membrane. Sometimes an enzyme comes and cleaves it, and now it's soluble TNF. So membrane bound versus soluble TNF. The biology of what they do is completely opposite. So the soluble form is pro-inflammatory and it promotes demyelination and neurodegeneration. So what do? How do we block it, right? Now, people that took those drugs that I just told you about, you're blocking both sides because the drug doesn't distinguish the membrane bound versus the soluble. So what happens? they're prone to opportunistic infections. They can get TB. They had to hide during COVID because they were immunosuppressed because of this side. So the idea is, why don't we just target that side instead of both sides, right? So that we don't make people immunosuppressed. And this is why we think that TNF is like a Dr. Jekyll and Mr. Hyde. It's got a good side and a bad side if it's in excess, okay? And so when I was in biotech, I had the fortune of being able to develop this next generation TNF inhibitor, if you will. And this next generation TNF inhibitor does not neutralize or does not block the effect of the membrane-bound TNF that's good, okay? It only sees and interacts with the soluble form. So we're able to just block the right side of the pathway and leave the immune protective side untouched. And so there has been a lot of studies showing how this novel inhibitor, right, looks like TNF, but it doesn't cause inflammation. But when it sees TNF here in the 3 white eggs, this drug is basically able to sequester the bad TNF into these heterotrimers, and they can't bind the receptors in blue. So it basically sops it up like a sponge. And the good arm is untouched. So you're not immunosuppressed. And you can give these to mice and then give them an infection like listeria, for instance. And the ones that have the conventional anti-TNF drugs heal over. The ones that have the soluble TNF specific inhibitor, XPro, do not. So we have evidence that it's not immunosuppressive, but it's able to combat inflammation. There's been now over 75 publications, not from my lab, but other labs as well, that show you that this is one way to decrease inflammation without immunosuppressing. So where is this compound now? So the Alzheimer's Association funded the company that licensed these inhibitors. I didn't have anything to do with the trial, but it's called Park the Cloud. And what they did is this very small trial of 18 people in Australia with either MCI or Alzheimer's. And they wanted to see if giving them this drug would lower their inflammation. So what did they do? They recruited people with inflammation. They didn't really care whether they were amyloid positive or tau positive. They needed to get people with inflammation. So they looked for CRP levels. Any doctor's office can do it. The sedimentation rate, the hemoglobin A1C, and people who were ApoE4, which tend to be more inflamed and at higher risk for Alzheimer's disease. So this trial was weekly administration of the drug subcutaneously for 3 months. And then they looked in the spinal fluid and they did MRI, right? So MRI, spinal fluid, and this is what happened. There were 2 doses. So this is open label, right? No placebo control. That happens in phase 2. But in the phase 1, they found that about 27 factors of inflammation were decreased after 12 weeks of administration. And the MRI showed that there was decreased free water, is what they call it. It's a measure of inflammation that you can do by MRI. Nobody needs to poke you to get a lumbar puncture anymore. The reason is the correlation between the CSF inflammation and the MRI inflammation was really good, 0.75. So in the phase 2, they're not going to collect CSF. They're just going to do MRI, less invasive. And here's what the evidence showed. Again, small group. This is the first half of the patients, N of 6. They looked at white matter fiber density. So these are your axons, your white matter tracks that carry the impulses, right? And after 3 months, they found an increase in the apparent fiber density. That means that they were fatter and more robust cables, if you will, okay? And compared to a group that didn't have that intervention in the purple, their apparent fiber density would decline over 2 years. And with the soluble TNF inhibitor, it improved about 10%. So There's now overwhelming evidence that what we see in the mice and monkeys and other models is translating into the clinic, at least with a small group of people that have inflammation, where you can lower the inflammation. And the next important step is, do you get any clinical improvement, right? Is their cognition any better? And you have to have a placebo-controlled. And so this phase 2 is enrolling now. Hopefully we will be able to test this in a Parkinson's population or any other neurodegenerative disease that has inflammation as its hallmark. Okay, so here's a take-home message. Immune cell aging and chronic inflammation resulting from this gene by environment interaction represents the perfect storm for Parkinson's development. We have now good evidence that gut health is important for brain health, and that the aggregation that appears to be important for the motor symptoms in the brain may start in the gut, right? The jury's still out. A lot of people are convinced that's how it is, but I think we really need to nail it down. And the problem is it takes so long. Like, who's going to run a 20-week trial, a 20-year trial? But what I think is important is that people who have these autoimmune diseases, they have an intervention. And the intervention is to quench their inflammation. And as a group, they have decreased risk for these diseases. The data is the same for Alzheimer's disease and for Parkinson's. If you are on these drugs for, a decade or so, you have less chance of developing Alzheimer's or Parkinson's when you're older. There's similar data with ibuprofen, but it's really interesting that not all the studies with ibuprofen show that there's a protective effect. And what's interesting to me is that studies where people took ibuprofen in middle age show protection. Those that were taking it right before their hip replacement, no protection. So that suggests there's a window of opportunity during which we need to identify people at risk and intervene with very safe drugs, good diet, physical activity in that period. So in humans, lifestyle choices, diet, exercise are all likely to affect the G times E interplay to impact gut and blood-brain barrier permeability, both of which will influence brain inflammation and your risk for PD, and I dare say probably Alzheimer's as well. So mitigating chronic inflammation and chronic systemic inflammatory diseases may reduce the risk for PD and other neurodegenerative disorders and its progression. And so to give credit where credit is due, much of this work was done at Emory before we moved in 2019. We have a great group of people now. This is a slightly outdated photograph, but we operate as a team. We believe that science is a team sport. And so a lot of different people are working on this problem. Some are working on Alzheimer's, some are working on Parkinson's, but the theme is the same. How can we understand the immune system's role in protecting you or predisposing you to these age-related diseases? And I think the final take-home message is that what they told us was good for your heart and to prevent cancer is the same that is good for brain health. I think neuroscientists were just a little slow on the uptake and thought that the brain was isolated and immune privileged, but I'm here to tell you it is not. And if you have taken care of your heart and done what you can to fight cancer, you will probably also be benefiting your brain. I'm happy to answer questions. There's one right there in the second row.
Hi, thanks very much. We've had talks before about oral health, and that seemed related to this. Could you comment, please?
Very much so. So there's good epidemiological data, again, that periodontitis is a risk factor. for Alzheimer's, in particular for Alzheimer's. And that could be because there's just more Alzheimer's and there's a lot of periodontitis, right? I'm not saying it doesn't contribute to PD, but the inflammation that you have chronically in your mouth is definitely affecting your brain. And so I think every barrier site, we need to do what we can to prevent the inflammation. We don't understand how the immune cells that become activated in the brain get those signals. They could be coming from the gut, they could be coming from your lungs, they could be coming from your mouth. So yes, periodontitis is a risk factor for dementia.
Dr. Tansey, we have 2 questions up on the screen on chat.
Okay.
Can you see them? Do you recommend probiotics? And
Yes.
our
Okay.
favorite, is there a minimum daily amount of red wine?
Of wine. Right. So probiotics, I'm going to say that I don't recommend against or for probiotics in terms of wanting to decrease your chances for these diseases. I think probiotics are great if they make you feel better, if they make you regular, if they make you feel good so that you want to walk around and be socially active and engaged. That is great. I just wouldn't spend too much money on them because, as I said before, there is no evidence scientifically that it can arrest or prevent the diseases. But we do have connections between gut inflammation and the risk for these diseases. So you could infer that, good gut health, however you get there, is a good thing. But there's no evidence that by taking them, you will be able to prevent these diseases. Not yet. We're working on it. We want to identify which bacteria are the bad ones, which ones are the good ones, and then maybe put those in a pill. Red wine. Right. So red wine, there's been a lot of studies with something called resveratrol, right, which is the idea that by consuming red wine, you're able to get better brain health and maybe heart health. Say that again? Yeah. So it's probably true, but you would have to consume huge amounts of it. And you don't want to do that, right? You'll have other problems with your liver.
Whatever it takes.
Yeah, whatever it takes. So I would say that, there's been some studies trying to link alcohol with protection and other studies saying that they give you increased risk. I really don't think that you should put too much stake in them. Just, moderation is good. And there's no good evidence that it's going to cause anything, right? It's going to if it makes you feel good, if it makes you sleep better, great. But in terms of the resveratrol, which is the active ingredient, right? You would have to consume trucks of it to get it to be protected. Same with curcumin and other things that have been tried.
Hi. To determine your level of inflammation,
I'm
can
sorry,
you
we're
recommend some tests?
a little bit louder
that
through
should
your
be
mask.
done? To determine your level of inflammation, can you recommend some different tests that should be done?
Yeah, that's a good question. So, whenever you have lab work at your doctor's office, they almost always check CRP levels because C-reactive protein is on the panel. And there's no number that will definitively tell you whether you're at risk, but there's definitely a threshold for cardiac risk if you have too high CRP. So we're talking below cardiac risk levels and above non-inflamed young people. Okay, so it gives you a huge range. And if you really want to know if your sedimentation rate, that tells you whether there's an infection. Your hemoglobin A1C, which is, pre-diabetes, those are all good indicators. So if you keep an eye on your numbers, I think you'll gain some information. The problem is that there's no validated biomarker or threshold cutoff that we can give people to say, above this, you're at risk, below this, you're not. So in general, keeping inflammation down in your body is the best way to go. And I always say that the best anti-inflammatory is physical exercise. Walking 20 minutes every day, break a sweat. It's different from different people, different paces. It doesn't matter. Whatever it takes for you to break a sweat, 20 minutes is what you need to do. Okay. Do you have studies continuing to research this time that want? Yes, we have several studies that we would love to have you participate in if you like. Some of them have to do with understanding, is there really a relationship between IBD and Parkinson's? So we are actively recruiting people with Parkinson's, people with IBD, and controls. And in fact, we're really needing controls. We don't have enough controls. We now have a good number of Parkinson's patients. We're almost there on the IBD from the GI clinic here, but the controls, we only have 15 and we need about 40 in order to be able to really interpret it. And for these studies, you don't have to be a spouse, but you can be if you want. So those involve blood, stool, and a short questionnaire. And we provide, I think, $100, $50 at the blood, and $50 after we get the stool and the colonic biopsy. And the colonic biopsy is not like a procedure that is invasive in the sense that it's not an endoscopy. They do it kind of like a rectal exam. So it's very fast. You don't have to prep for it. You don't have to drink the Go Lightly at all. That's the worst part. So it's an unprepped sigmoidoscopy. And that's so that we can get a small biopsy and we can look at the molecular changes in your gut, whether you have PD, IBD, or a control. So those are some of the studies we're enrolling for. We are also enrolling for what we call the most common form of genetic Parkinson's, which is the LRRK2 mutation. So if you have Ashkenazi Jewish ancestry in your family, there is a mutation that appears more commonly in that population, that if you end up with Parkinson's disease, you may have that, right? And so if you know that you have a LRRK2 mutation or have done 23andMe and have been told that, we're also enrolling that particular group of people. And we've gone to synagogues recently to try to recruit more subjects because we're just starting that part.
Dr. Tansey, if you want to share any of the contact information for the study with me, I'm happy to send it out to this
Wonderful.
group.
I will give it to Julie. Yes.
I was wondering, with some of the information coming about for various viruses and organisms like herpes and that contributing to neurodegenerative disorders, in addition to cytokines and proteins that are traveling up and down the vagus, do any of the microbiota organisms themselves travel up into the brain?
That's a great question. There's a group of people, mostly out of Harvard, that have been working on this problem. Rudy Tansey, different spelling of Tansey, with a Z, not an S, and his colleagues. And their idea is that the reason the plaques form in the brain is to trap bacteria, like a net. And I don't know how we're ever going to prove that or test it. But there are studies trying to figure out if the bacteria that some investigators find in an autopsy is there because it got contaminated somehow, or if it really got there through your body while you were still alive. So there is a connection. You probably read the paper or heard about it in the news, that Epstein-Barr virus was thought to be the primary cause or increased your risk for multiple sclerosis, right? The problem with those studies is that they didn't look at the genetics of those individuals, right? Epstein-Barr virus, almost all of us got it, right? It's mono when we were in high school, right? So almost all of us had some kind of mono, but we don't have, MS, right? And so what you have to think about is it may be required, but it's not sufficient to give you the disease. So something else is interacting with those viruses to increase your lifetime risk for those diseases. COVID-19, what is it going to do? Well, there's good evidence that people with long COVID appear to have more inflammation just in their bodies and potentially in their brain. If that is true, one might predict that if they don't get that inflammation under check, that may contribute over many years to one of these age-related neurodegenerative diseases, much like we know that encephalitis, right, with the Spanish flu, caused an epidemic, if you will, of Parkinsonism. It's that movie Awakenings with De Niro. Wonderful story. Oliver Sacks was the neurologist. And so there's a concern, but there's no evidence yet that anything like COVID or long COVID is contributing to these diseases. Because they take so long to develop, it's going to be one of these natural history experiments that we're just going to have to see. Yeah, it's not thought that they can, but substances and metabolites that get out may be there, and we just don't know where to look, right? So the bugs, I think, are not likely to do it, but the metabolites and the things they make, they also make, they're super clever, they make proteins that look like synuclein. They're called bacterial amyloids, and they're actually thought to be important for fighting certain other infections. So as Pushpa said at the beginning, there are many reasons to believe that bacteria are super critical to our existence, right? If you put a mouse in a germ-free facility, their brains are all messed up, right? And we need certain microbes. We want to just have them in balance, and we want to understand where that balance is lost as you age, where the balance is lost when you make certain lifestyle choices and diet, and we want to be able to identify people that are at higher risk to be able to tell them, here's your genetics, you can't change those, but your environment and your lifestyle choices, you probably can.
Okay, and our last question here, go ahead.
I actually have two questions. I hope I can get them in. This is a fascinating lecture.
Speak hold the microphone right like this.
Okay, fascinating lecture. Can you recommend any books that we can read to follow up on this? And the other is, I understand that there's no special diet, but are there some types of foods that are definitely negative toward these things?
Great question. So if you want to know more about the microbiome, there are several books that have been written that are in the bestsellers list. Rob Knight, who runs the Microbiome Project, they're collecting stool from every American if they possibly can. Rob Knight wrote one of those books. I can't remember the name, but it's Knight, Rob. And Michael Okun, the chair of neurology here at UF, wrote Living with Parkinson's, and that's a really good book that just talks about risk factors and various things. So I would recommend that one. There's in terms of the foods, I would say that there have been studies that suggest that foods very high in saturated fat are not great because they do create inflammation, at least in animal models of high fat, high fructose diet. So we've done some studies where we take a mouse that has a genetic predisposition for amyloid forming, and when we put it in a high fat, high fructose diet, that accelerates, right? And the immune cells in the blood go into the brain, and we gave them the Expro drug, and that seems to prevent it and reverse it, which is great, which is part of the reason that it was taken into clinical trials in Alzheimer's. But I would say that, fresh fruits and vegetables, same diet that the American Heart Association recommends for your heart is exactly what I would follow. And then physical activity, right? The walking is really important. The sleep is really important because these aggregated substances are removed while you're sleeping. And if you don't sleep, then you don't remove them. So sleep is really critical to the process of keeping your brain healthy. Yes.
Thank you so much. This has been a fantastic start.
Thank you for the invitation, and I hope to see you again soon.
I hope so, yes. Our next talk in this series next Monday is why do we continue to eat foods that we know are bad for us? And it's a natural progression to that talk. And the speaker is from Pennsylvania, and he will be talking on Zoom. So he will not be present here personally, but I still hope you will come and we'll have the same format. Thank you all for coming. It's very nice.